Now we'll talk about graft versus host reaction or graft versus host disease. What is graft versus host disease? This disease results from when donor lymphocytes attack the graft of the recipient. Immune response usually recognize and destroy transplanted organ. However, the situation is different when competent immune cells are transplanted into a recipient. This can happen during bone marrow transplantation when normal donor T cells may be infused into the recipient. In such circumstances, the T cells can recognize the MHC molecules of the recipient as foreign and produce an immune response against the recipient. This is known as graft versus host disease. In this disease, graft versus host disease, it can be very lethal, causing damage in particular to the skin and gut. It can be demonstrated in animal models by transfer of bone marrow to irradiated recipient animals. It can be, however, avoided by careful matching of the donor and the recipient, by removal of the T cells from the graft, and immunosuppression. Okay, so what is graft versus host disease? This is a disease caused by when donor lymphocytes attack the graft of recipient. Okay, so what's the hypothesis or mechanism behind graft versus host disease? So here we will take 3 strain of mouse. This is strain A, strain B, and strain C, and we'll X-ray irradiate strain C to make it immunosuppressed. Okay, so immunocompetent cells from a donor of the type A are injected into immunosuppressed, that is X-irradiated host of type C, or a normal A cross B mice, that is the F1 recipient of donor A and B. The immunosuppressed individual is unable to reject the cells, this one. So strain C will be unable to reject the graft from A. However, F1 animal is fully tolerant to parental type A cells, so it will accept the graft from this one. Okay, because donor A is one of this mice parent. So what happened then? In both cases, the donor cell recognize the foreign tissue types B or C of the recipient. They divide and react against the recipient tissue cells and recruit large number of host cells to inflammatory sites. Very often, this process leads to the death of the recipient. So this, as this mouse is immunosuppressed, it will gather a large amount of recipient tissue cells and recruit the host cell, which leads to inflammatory reaction, and this inflammatory reaction can ultimately lead to the death of the mouse. However, this mouse is tolerant to this one, cells from this one. Okay, so this mouse will not show any kind of immuno reaction. It will show tolerance towards the graft of donor A. In organ transplantation, strategies used to avoid or delay rejection by using immunosuppressive drugs and minimizing the extent of the specific allogenic reaction. Although rejection can occur, they are usually kept in check by the drug, so the lasting damage is minimized. The commonest cocktail of drugs used for kidney transplant patients involve 3 agents, each of which has a distinct mode of reaction. So a drug that can inhibit T cell activation, anti-proliferative drug, and anti-inflammatory agents. So what are the immunosuppressive drugs? 6 mercaptopurine, azathioprine, and myophenolic acid, that is MPA. These are anti-proliferative drugs. They disrupt the synthesis of DNA or RNA and cell division. They prevent cell proliferation. Then cyclosporin, FK506, and rapamycin. They inhibit T cell activation. Cyclosporin and FK506 affect cytokine production, and rapamycin interferes with the signaling through the interleukin 2 receptor. Corticosteroids, they inhibit pro-inflammatory cytokine secretion, reduce inflammation, and inhibit adhesion molecule excretion, leading to reduced inflammatory cell migration. These are the anti-inflammatory drugs.